Yeah, put it right here. Where was it? I'm gonna try the light.
I don't know how I feel about that.
Okay,
That's like
are we ready to do our primary exercise for the evening? Hopefully the answer is yes. So last time we began our staining operation, we did a direct stain and an indirect stain, but you didn't get to look at either one of them. Hopefully you guys got them all put together. Tonight we're going to do the gram stain, and then you will have time to look over these because it's only, well, it's not quite 8:30. We go until 10:05. You should have plenty of time. So this is staining of microorganisms, gram stain. The exercise is on page 63. So the gram stain. This is a method of staining. It was developed by Hans Christian Gram, a Danish physician, in about 1884. What it does is divide medically significant or clinically significant bacteria into two categories. The categories are gram-positive and gram-negative. It works pretty well for clinical varieties. Turns out it doesn't work all that well for environmental varieties, but it's still used, and it's used extensively in the clinical setting. It has somewhat variation in application, depending on whose lab book you're looking in. Always there's going to be alcohol involved. Sometimes there's acetone involved. Our particular gram stain involves a decolorizing reagent that is an acetone alcohol mix. It's quick. It's faster acting than a plain alcohol solution. So there are slight variations on a theme. This is an example of a direct stain. So if you recall last time, our direct stain was cheek cells. You haven't had a chance to look at those yet. But a direct stain colors the cells. The background doesn't take up the color, and as a consequence, the background is going to appear white when you're looking with a light microscope. This is also a differential stain. This is a new category. A differential stain is one that causes cells to look different, even though you treat them all the same way. They're going to end up looking different because they are different. In the gram stain, we should have a set of reagents. One thing I told you guys about the gram stain reagents, and that is keep them in the box in the right order. The gram stain involves crystal violet, Brown's iodine, a decolorizing reagent, and saffron. Those four reagents should stay in this box. Periodically, we will have other reagents in the green trays, but make sure your gram stain set in the box is right. If you keep the reagents in the right order, then it's easy to remember how to do a gram stain. The other thing that goes in this box is your immersion layer. So it's differential. We're going to treat all the cells with all four reagents. Some of them are going to come out pink, and some of them are going to come out purple. And the reason they come out looking different is because they are different. So it is a differential stain. Gram-positive bacteria are dark and purple. Gram-negative bacteria are light and pink. The shapes that you see here has nothing to do with the stain. There can be gram-positive cocci and bacilli. There can be gram-negative cocci and bacilli. In order to understand how the gram stain works, we have to look at the composition of the wall. So I touched on this a little bit in lecture, but here we're going to talk about the composition of the wall in greater detail. So remember, we're talking about the cell wall. This is outside the cell membrane on both gram-positive and gram-negative. When we're looking at bacteria that are referred to as your ordinary gram-positive and gram-negative bacteria, which that applies to most clinical strains of bacteria, their wall, that is the rigid portion of their wall, contains peptidoglycan. As I said in lecture, peptidoglycan is a complex material. It is unique to bacteria. Peptidoglycan is found in bacterial cell walls and no other type of cell. To get an appreciation for what peptidoglycan is like, consider a phone book. This becomes a problematic thing to ask students to consider a phone book because now everybody has cell phones. And it's like, really? Do you know what a phone book is?
Yes.
So they do still exist. Oftentimes they're just a collection of yellow pages and they're a bunch of advertisements. Typically, phone books have relatively thin paper, and the thickness of the book is dependent upon how many pages you have. So peptidoglycan is somewhat like that. You can have very thin peptidoglycan, which means you have very few pages in your book, or you can have thick peptidoglycan, which means you have many, many, many pages in your book. Well, the page is one layer, which tells you peptidoglycan is a layered material. So here's my phone book, and I'm gonna say that the phone book, the pages of the phone book represent multiple layers of peptidoglycan. This is kind of a medium-sized phone book, and then I'm gonna try to wrap it around the cell. You can see it doesn't behave very well, but you get the general idea. Peptidoglycan is multiple layers wrapped around the cell. If the cell is gram-positive, you have a very thick, many, many layers of peptidoglycan. If they're gram-negative, you have few layers. Peptidoglycan is thin. Yeah, now visualize taking one page and just taking out a tiny little section, because we want to look at the actual molecular composition of peptidoglycan in that tiny section. So each peptidoglycan layer is composed of some polysaccharides. The polysaccharides involved are N-acetylmuramic acid and N-acetylglucosamine. Used to be nobody knew what glucosamine was, and then you start seeing glucosamine on billboards out there along the freeway. Glucosamine is a substance that more people recognize, but it is part of peptidoglycan. As far as I know, the N-acetylmuramic acid is only found in peptidoglycan, not so much the glucosamine. Anyway, these two sugars, N-acetylglucosamine and N-acetylmuramic acid, form polysaccharide chains. So we have alternating sugars. In this diagram, remember, we're looking at a single page of the phone book, one layer of peptidoglycan, and we've torn a little section out of that one page. So we're looking at just a little piece of it. These chains extend out and around the cell and all the way back and around. We're just looking at a tiny piece of one layer. We have alternating sugars here forming these polysaccharide chains. Now, associated with the polysaccharides are short peptide chains. So peptidoglycan, glycan refers to the carbohydrate, peptido refers to the peptide chains. These are amino acids connected together. There are pentapeptide bridges and short peptide chains hanging down. So we see our short peptide chains hanging down under the N-acetylmuramic acid. Each one of those has a little short peptide chain hanging down. This peptide chain would connect to the next sugar, the sugar of the layer underneath. There would be another chain coming down from the N-acetylmuramic acid of the layer above, and it would connect down this way. So the chains are connected between the layers by these short, or the chains of sugars are connected to the next layer by these short chains of amino acids. And then these are pentapeptide cross bridges connecting this chain of amino acids to this one so that it's holding this whole thing together. Again, this is one tiny section of one page, and all of the pages are going to be connected together by these chains of peptides. So they're all really connected to one another. What that tells you is peptidoglycan is huge in terms of molecular size, because it's these big long chains of polysaccharides, and it's all interconnected by peptide bridges, peptides. What's interesting about the amino acids that are associated with these peptide chains, some of them are never found in protein. I don't know how many of you have had organic chemistry where maybe you had to learn all the different amino acids that are in proteins, but I've mentioned to you there are about 20 types. So you have leucine and isoleucine and alanine and phenylalanine and threonine and so forth. You have a certain number of amino acids. The peptide chains in bacteria, some have some amino acids that are never found in proteins. They're weird. So we have diaminophenolic acid and diaminobutyric acid. These are amino acids never found in proteins, but they show up in peptidoglycan. So this is kind of another unique feature of peptidoglycan. It can hold some very strange amino acids, and there are groups of researchers now that are trying to artificially build brand new amino acids and then give them to bacteria and say, here, can you use that? Can you put that in your wall? And sometimes they can. The other thing about the amino acids that are found in peptidoglycan is some of them are backward, that is, they're mirror image molecules. So if you look at a peptidoglycan, if you were to set it in front of a mirror, you would see that the mirror image is backward. The amino acids that occur in proteins within living organisms are all L form. It'd be handy to tell you that the L stands for living. It doesn't, but it helps because you can say, oh, they're L form, living. Some of the ones in peptidoglycan are D form. It doesn't mean they're deformed. It means they're mirror image molecules. They're the D form instead of the L form. And there you can see the mirror image amino acids rotating around. So that's significant. Now we're looking at a diagrammatic representation of the gram-positive wall and the gram-negative wall. And what we're doing basically is looking at the gram-positive cell. The yellow layer here is cell membrane, and then we have a very thick layer of peptidoglycan. Imagine that extended downward. Here's the cell membrane, and here's the peptidoglycan. It's a very thick layer. On our gram-negative cell, we have cell membrane, and then a thin layer of peptidoglycan, and then an outer membrane. So here's our cell membrane, thin peptidoglycan, outer membrane. This outer membrane is kind of weird. It doesn't actually have the same composition as the cell membrane, and it has a whole bunch of these polysaccharide tails sticking out of it. The gram-positive wall also has teichoic acid in it, and in the diagram that I showed you earlier, these appeared to be kind of strings. That structure is obviously not very accurate. So the gram-positive has a very thick peptidoglycan wall. Gram-negative has a thin layer of peptidoglycan and 2 layers of membrane. That outer membrane in the gram-negative wall has complex composition. We know the composition of the cell membrane. It's about it's the prokaryotic we're talking about, so it's about 60-40 protein to lipid. But we talked about the composition of the proteins, integral, peripheral. We talked about the composition of the lipids. Most of them are phospholipids. This membrane is a little bit different. So you still have proteins and phospholipids, but you've also got some lipoproteins, and you've got these lipopolysaccharides. You've got these long carbohydrate chains sticking out, sometimes very long. What's significant about the lipopolysaccharide, oftentimes abbreviated LPS, is these things are toxic to mammalian cells. So sometimes when you have bacteria get into the bloodstream of a patient, even if the bacteria are killed by giving the patient an antibiotic, when the bacteria die, they release these lipopolysaccharides, and these things can cause damage even though the bacteria are dead. So it is fairly significant. So these are diagrammatic representations of gram-positive wall. This is taken from a textbook. Unfortunately, it's upside down relative to my diagram. So again, we have cell membrane. In this case, the periplasmic space is this little bit of space between the membrane and the wall. And here we have multiple layers of peptidoglycan. We have some teichoic acid in here. Most important aspect is the peptidoglycan layer is really thick. We compare that to the gram-negative wall. Again, this diagram is upside down relative to the one that I showed you. So here's our plasma membrane or cell membrane. Here's the thin peptidoglycan. Here's the outer membrane with these lipopolysaccharides, polysaccharides extending outward. There's lipoproteins connecting the peptidoglycan to that outer membrane, and the periplasmic space is the whole space between the membranes, such that the peptidoglycan is in it. If this cell had periplasmic flagella, they would be connected to this layer and extending into this space. So they'd be inside the outer membrane. It doesn't. Now, when we do a gram stain, we're taking advantage of the fact that these walls are in fact different. The steps in the gram stain involve, first, the application of a primary stain. Our primary stain is crystal violet. When we did the cheek cell stain with crystal violet, you were supposed to stain for 10 seconds. 10 seconds is plenty to get a lot of purple color on cheek cells. But for the gram stain, we apply this color for 60 seconds. We're gonna color everything purple. The next thing we do is apply a mordant. Oh, this is talking about why does the color bind to the cells? Remember that crystal violet is a basic stain. Basic stains have their color associated with cations. The cations are going to try to get to the negatively charged membrane. With a gram-positive cell, they're going to go into the peptidoglycan in order to get to the membrane. With a gram-negative cell, the outer membrane, there's a negative layer outside, so the dye doesn't actually get in. So our gram-positives are going to be stained more completely than the gram-negatives. The next thing we do is put on a mordant. That's our grams iodine. We're going to apply this also for 60 seconds. The mordant causes the crystal violet, well, it actually crystallizes with it. So it's supposed to bind the color to the wall. Doesn't change the color of the cells usually. I say that and I think sometimes it does. Sometimes it makes them darker. They'll be purple, but they may be so dark they almost look black after you put the mordant on. This binding of color is going to happen throughout the peptidoglycan on the gram-positive, but it happens more or less on the surface of the outer membrane on the gram-negative. Can't get in. You can sometimes see the crystals that form. If you put crystal violet on, you don't rinse thoroughly before you put the grams iodine, the surface of your slide will be covered with crystals, and they're these spiny looking things are all over the place. So try not to do that. The third thing we apply is a decolorizing reagent. The decolorizing reagent we use in this laboratory is acetone alcohol. So it is very rapid acting. 70% ethanol and 30% acetone. Acetone is fingernail polish remover, so if you have really fancy nails, watch out for our decolorizing reagent. It will mess them up. The decolorizing is designed to take the color out of the gram-negative cells quickly. Why? Because the color never really got in. It's sitting on the surface of the membrane. It's pretty easy to remove that color. For the gram-positives, the color got into the wall. It's going to be hard to get out. At the end of the decolorizing step, your gram-negative cells should be clear, colorless, and your gram-positive cells will still be purple. If you do this wrong, you can decolorize everything. So there's a specific method for applying the decolorizing reagent. Anyway, the last thing we do is put on a counterstain because we want those gram-negative cells to show. We counterstain with safranin, and safranin is going to color the gram-negative cells pink. So put that on there for 60 seconds, and you should see gram-positive purple and gram-negative pink. If you do it right. And the safranin is lighter than the crystal violet also, so your gram-negative cells will appear lighter, definitely pink as opposed to red. So this is an illustration. It's on the website. You might see something very like this in one of your preparations tonight. This is showing Bacillus cereus. The bacillus are on slants in the white trays that are over here. So Bacillus cereus is a large gram-positive type bacteria. If they are making spores, the spores will show up as white spots. So these are endospores. Also in this illustration are little bitty cells. These are Serratia marcescens. Serratia marcescens are gram-negative. They're very small. So a lot of you are having trouble getting isolated colonies with Serratia marcescens. It's because they're tiny, and every one of them wants to grow when you put them on a nutrient agar plate. They're very growy. Okay, so here's the procedure for doing a gram stain, and I'm going to put this on the board after I go through this. So on a single glass slide, I'm going to ask you guys to prepare 4 smears. Remember, keep your smears small, and you want them to be uniform, a few cells in liquid. We're going to set up a slide where we have a gram-positive culture at one end, and we're going to make that in the sign of a plus sign. In the sign of a plus sign? Just make it a plus sign. Then we're going to have a gram-negative culture. We're going to make that a minus sign. So here's our plus sign, here's our minus sign. In between, we're going to have a mix, which is just going to be kind of a ball. And we're also going to put our morphological unknown on there. which is going to be in the shape of a U. The reason for doing this is so that you can tell these things apart when you go to look at them. It sounds kind of complicated, but it's like, really, you can do this. Keep your smears small. Don't cover the entire slide. So you make your smears, they look like that. This tells you exactly which cultures we're using. So we have 2 gram-positive cultures. They are Staphylococcus aureus and Bacillus cereus. They are in the trays. So each table has 4 tubes, 2 of Staphylococcus, 2 of Bacillus. These are 24-hour-old cultures, so they should be in good shape. The problem with gram-positive cultures is they will lose their stain quality over time, so we want them to be young. We're going to put Staph in the plus sign. We're going to put Bacillus in the mixed smear. Then we have 2 gram-negative cultures. These are Serratia marcescens and Klebsiella. Serratia, we've got Serratia all over the laboratory because a lot of people streak Serratia marcescens for their pure culture plates. Hopefully you still have those. So that's the red is Serratia marcescens. The Klebsiella may be a little bit harder to find. There were a couple of Klebsiella pure cultures, but if you look on your Mix A plate, which I told you guys to keep, the bigger columns that are kind of slimy, wet looking, that's Klebsiella. So the Klebsiella goes in the minus sign, the Serratia goes in the mix with the Bacillus. And then again, your morphological unknown is at the end. Notice that the smears appear whitish against the dark background. So do not put your slide on a piece of paper and you can't see what you're doing. So we make our smears. Here's showing Serratia and Bacillus on the Mix B plate. Serratia is the red one. You can get it from a lot of different sources. The Bacillus, use the slant, please. If you use those old Bacillus cultures, this gram stain quality won't be good. Klebsiella, again, the Klebsiella colonies are the big slimy looking ones on the Mix A plate. Big slimy Klebsiella. The little white ones are Staph, but don't use the Staph off your Mix A plate. Use the Staph from the slants, because these are younger cultures. So again, the Staphylococcus and Bacillus, please use the slant cultures. These are young. They should give us good positive, good purple gram positives. So after you've made your smears, you air dry them and then you heat fix. So remember, pass them through the flame of the Bunsen burner 3 times, relatively slowly, like this. Once, twice, 3 times. Now, if you can only hold on to one end of the slide and you heat fix the 2 smears at one end, then turn it around and heat fix the 2 smears at the other. But don't hold it in there for a long time. We don't want to charbroil these guys. Your reagents are in this order. They used to have marks on the tops of the caps, and that was supposed to help students not mix up the caps. Please don't mix up the caps. The colors didn't work. Telling you probably won't work either, but. So, how do you do this stain? Once you've got your smears on the slide, they've been air dried, they've been heat fixed, you put them down on the stain rack over the sink, and you cover that smear with crystal violet. Be careful with the crystal violet. Remember, this stuff is exceedingly messy. Try not to get it all over the place. So you put that on there, let it act for 60 seconds. Then you're going to rinse. Again, hold that slide down in the sink at an angle, rinse off the excess stain. The stain is stuck to the cells because crystal violet is a basic stain. It's going to bind to the negative surface of the cells. So we rinse it off. Then we put on Graham's iodine. We're going to let that sit on there for 60 seconds. Just pour it on there, cover the entire thing. Let that sit 60 seconds. Then you're going to rinse that off. And then you're going to decolorize. Now the decolorizing is a tricky part of the procedure. This is really what makes the difference. You have to hold the slide so that it's in a kind of a vertical position. I'm going to get a slide here. You want to hold it like this. You can see that? This way, not this way, this way. Not that way, this way. And if you can, have something white behind it. That's why we have this white conduit. Unfortunately, it doesn't go all the way to the end. You want to be able to see your smears, and you want to hold this over a sink because you're going to apply the decolorizing reagent, and you're going to continue to apply it as long as the color flows. When the color stops moving, rinse it. So typically when I do decolorizing, I want the water running. So I've got my tap going, I'm holding my slide. Apply the decolorizing reagent, see the color flow? As long as it keeps flowing, keep putting it on. When it stops flowing, rinse. If your smears are chunky, it's gonna be hard to decolorize accurately. That's why you want a uniform smear. You're gonna put that stuff on, watch the color flow. When it stops flowing, rinse, you're done. Rinse it thoroughly because you wanna get all that decolorizing reagent off. With that really got out of focus, didn't it? Use caution. Don't decolorize too much, because if you do, you'll take the color out of everything. You can over decolorize and then your gram-positive bacteria won't be purple. And we want them to be purple. You don't want to see everything clear. So then you put on the counterstain, safranin. Again, 60 seconds, just cover the entire slide. And you rinse off that. You're ready to air dry the preparation, and then you can observe it. I want you to observe first with the 10x. Once you have looked, and do one smear at a time, because when you put the immersion oil on, it tends to sit there as a convex surface, but gradually it will flatten out. So you don't put oil on all of your smears and then start looking. Put oil on one smear, look at it. Then put oil on the next smear, look at it. And just focus with 10x and then look at them with 100. Don't use the 45. You're going to get refraction with the 45x and you can't do anything about it. So focus with 10, then go directly to oil. You're going to record what you see. So you have places to draw these. When you draw them, try to make your cells big enough that they look like cells, not dots and specks. Think about the pictures in the window cases. You want to draw cocci that are distinctly spherical, ball-shaped, like marbles. You want to draw bacilli that are rods. Think hot dog or Myconites if you want them smaller, but definitely three-dimensional. So you're supposed to record your data. Data is what you see. You can draw pictures. You can write out verbal descriptions if you want to, but pictures are easier. Data is what you see. Purple cell, pink cell, data is what you see. You see purple cells, right, I see purple bacilli. I see purple cocci. Also record your results. Your results are gram-positive versus gram-negative. If they were purple, they're gram-positive. If they were pink, they're gram-negative. And then your conclusion, what kind of a wall do they have? If they were purple, they were gram-positive, they have a thick peptidoglycan wall. If they were pink, they're gram-negative, they have a thin peptidoglycan wall. So you want to record those three aspects. You're practicing with the mix, the positive, the negative that we know, and then you look at your morphological unknown and make sure that you can record all that information about it. This is talking about the KOH test, which is another way of determining wall composition without staining. This works pretty well, and sometimes works better than a gram stain. Turns out that the cells that have thin peptidoglycan, that peptidoglycan will break when you expose the cells to 3% potassium hydroxide. And when the cells break, the DNA comes out and mixes with the potassium hydroxide, and it forms this slimy goo. It looks like snot. Really, you stir it around, it becomes very viscous, you lift your loop up, and you get strings. So this is Serratia marcescens, gram-negative. See the viscosity? Slimy goo, snot-like. That means these cells have a thin peptidoglycan wall. It broke and the DNA got out. If you do that same procedure with a gram-positive type wall, that is thick peptidoglycan, it won't break. And you stir it around, the viscosity doesn't change, you won't get strings. So the KOH test is just another way to determine wall composition. Tends to confuse students. This is telling us that some types of bacteria are genetically identified as gram-positive, but when you stain them with a gram stain, they're pink. And for a long time, the reference volumes talking about bacteria kept talking about gram-negative bacilli. They really weren't gram-negative. They just didn't stain appropriately. So there's a bunch of bacillus that do this. That is, organisms in the genus Bacillus. Then you had to use the KOH test to really determine what kind of wall they had. So in this one, this says put 3 separate drops of potassium hydroxide. If you wanted to do this where you're comparing a positive that you know, a negative that you know, and your morphological unknown that you don't know, if you really feel that you need to do this, you can. Or you can work in groups and just do 3 unknowns on a slide. Each drop of KOH, potassium hydroxide, you put that on the slide, then you pick up a little bit of your bacteria. It needs to be a visible amount, a ball of about 2 millimeters in diameter, and you stir that into the potassium hydroxide. So again, a known gram-negative would be Serratia, a known gram-positive, either Staph or Bacillus, and then your unknown. If everybody does that, we're going to waste a lot of glass. So I prefer really that you do 3 unknowns on the same piece of glass. Forget doing the knowns. And this is a pretty fast reaction, so if the cultures are young, you're going to get a reaction in about 5 to 15 seconds. You get that slime right away. If they're older, it can take longer. Our cultures are not too old. So you want to record again with your KOH. You want to record your data, which is what you see, slime, no slime. Then your result. If you see slime, that means it is KOH positive, which means the culture is gram-negative. Got a thin peptidoglycan wall. So data is what you see, result is positive or negative KOH. The conclusion is, what kind of wall do they have? Thick peptidoglycan or thin peptidoglycan with an outer membrane. And your KOH results should match your gram stain. If they don't, the KOH is probably right. We have a culture that for years we thought was gram-negative because every time people stained it, they were pink. In fact, this culture, every time they're stained, they want to stain pink. They do not have a thin peptidoglycan wall. Okay, so I'm going to put the procedure on the board. Everybody have this stuff up here?